So, good morning.
Thank you, sir.
Can you hear me now?
Yes, sir, I am hearing you.
Okay. In our earlier class, we discussed about uh, nanotechnology in crops. Today, we shall discuss on nanotechnology for livestock
Let's talk.
healthcare.
Okay.
Livestock, poultry, they are very important for our nutritional security. Livestock production and other sectors of the livestock supply chain contributes significantly to regional communities and the overall global economy. Use of the state-of-the-art nanotechnology could be improved livestock healthcare uh, through the following aspects. Number one, developing a safe and efficient alternative uh, antibacterial nano formulation to antibiotic supplements in animal nutrition products. Nowadays in livestock and poultry, synthetic antibiotics have been used, are being used, and synthetic antibiotics are hazardous. Some of the residue are coming to our body through the product, through the food chain, and they are the big threat for antibiotic resistance. This is why antibiotic nano formulation, antibacterial nano formulation is one of the alternative to the antibiotics. Generating novel nanopesticides with improved safety and performance for the livestock pest control. Livestock has lots of pests, including the mosquito. So different parasites also there. So uh, nano pesticide can be alternative uh, to replace the synthetic and hazardous pesticide currently being used. Third, engineering nano-digestive enzymes to address small animal digestive problem. Not only livestock, all, mammals, uh, they have a big problem that digestion. A large proportion of the food or feed they are taking are unused and they are releasing through the stools. So increasing the uh, digestive uh, uh, digestion efficiency by using nano-digestive enzymes would be one of the important tool. Here you can see in case of bacteria, this is a bacteria, for example, Salmonella. If you apply nano pesticide or nano antibacterial formulation, and if it has some target specificity, then it will bind on the bacterial surface, not other parts, and it can control the bacteria selectively rather and high efficiently than the normal antibiotic. Normal antibiotic actually, and the antibiotics we are taking or livestock are taking, in all cases they are systemic. After taking, whole body is poisonous, and as a result we can see the effect, but it has serious stress to the body also. If you can design a nano antibacterial formulation or drug which selectively target the bacteria, that would be fantastic. So antibacterial nano formulation for livestock healthcare could be very important. Natural products, another approach is natural products are uh, our largest source of therapeutic molecules. Natural products, possibly you know that in the herbal medicine or medicine available in the plants and other uh, living organisms which have antimicrobial properties as well as growth promoting properties in the for human and other animals. They are called natural products. They are defined structure. For example, vitamin C is a natural product. So vitamin C, L-ascorbic acid, has a defined structure. And after discovery of L-ascorbic acid, scientists synthesized the L-ascorbic acid, and now you can get the tablet of vitamin C, which is very important for boosting your immunity, even against the COVID-19. So natural products are our largest source of therapeutic molecules. Over the past decade, sequencing bacterial genomes has revealed an immense reservoir of biosynthetic genes that have the potential to produce bioactive molecules. This is very interesting point. You should remember that the drug we are using now, modern drugs, most of them are derived from or discovered from the bacteria like streptomyces and other bacteria. So bacteria are the reservoir of biosynthetic genes of natural product, antibacterial compound, antiviral compound, or nematicidal compound, like that. So however, the majority of these genes appear to be inactive for unknown reasons, those biosynthetic genes. So method to activate the gene, these silent clusters would have a profound impact on drug discovery. The use of system biology means synthetic biology. You can synthesize the gene or design the gene for awakening silent gene clusters, because most of the genes are silent, they are not expressed. So if you can, by using system biology, if you can awake or that means able to express the silent gene cluster that are responsible for uh, bioactive compounds, can increase its bioactive compounds, that would be very interesting. That means expression of the silent gene which are not naturally expressed would be interesting way. Then you can get the targeted products. The approach combines high throughput omics. Omics means genomics, proteomics, metabolomics. There are lots of transcriptomics, omics approaches with synthetic biology. Omics approaches like transcriptomics. Transcriptomics approach, you can calculate the level of expression. Sometimes level of expression of a gene is poor, sometimes very high. So level of expression is very important. The application of this strategy promises to uncover the regulatory networks that activate silent gene clusters, while at the same time providing access to the vast array of cryptic molecules found in bacteria. Cryptic molecules means the molecules are not synthesized in abundant quantity. So if you can use the synthetic biology or system biology approach to increase the expression of silent gene, you can get those cryptic molecules as bioactive natural products. So it is hypothesized that, and it has already been proven in some cases, that synthetic biology approaches can enhance the expression of the silent gene and produce the cryptic molecules in large quantity. And then you can get those molecules as a rare, a drug candidate for veterinary or even human pathogen. So these are the approaches, novel approaches, which have been now considering by the scientists worldwide. Another is big chemicals. For example, biological gas in liquid production of chemicals, a system biology approach. System biology approach is now new approach. That is, you change the biosynthetic pathway or synthesize new, gene to, regulate the existing system in the, organism. The advancing waste gas fermentation process to enable the production of sustainable chemicals in an interesting targets. Langertech are world leaders in microbial gas fermentation and have produced ethanol at large scale. That means by using microorganism, if you can do the fermentation of biomasses, for example plant uh, residues, or plant how to say straw, or other residue, even in horticultural residue, fruit peel or other thing, all contains high amount of cellulose in their cell wall, and they contain carbon, hydrogen and oxygen. By using microbial fermentation, you can convert them into ethanol, and it is called bioethanol or biodiesel. Combining the Langertech process with system biology expertise to go beyond the ethanol to deliver new value-added products such as bioplastic. Bioplastic, plastic is a polyethylene or like that. So by using biomass, nowadays scientists develop technology to make the bioplastic. What is the advantage of bioplastic? Bioplastics are biodegradable, and this is a very big advantage because plastic or polythene is a big trouble for our environment. So if we can make replace them by bioplastic, that is converting biomass into the useful plastic, that would be very good. And perhaps you have heard about the bio polythene from the jute discovered by Dr. Mubarak Ahmed Khan in Bangladesh. So if you can make uh, polythene by utilizing the jute fiber, then that polythene would be biodegradable and very amazing technology. To achieve this, it aims to understand and uncover fundamental energy and metabolic limitations in the production microorganism. Achieving the aim will be a direct relevance to the, here Queensland Biofutures, they are working even in any country, any laboratory can do that. So in Queensland University, they are doing this type of research currently, but this type of research can be doing in any part of the world, and it is one of the emerging area for research in in nanobiotechnology. That means to make bioproduct utilizing the nano technologies. Here you can see the gas fermentation, Clostridium otutenongenum system biology approach, which Langertech is using. Here, these are the nanoparticles, this gray color and blue color are the Clostridium bacterium. They can break down the polymeric substances and make the nanoparticles, and this technique have been proven useful. System biology driven discovery and enhanced production of natural products. Natural products are our largest source of therapeutic, already I mentioned. Over past decade, sequencing bacterial genomes has revealed an immense reservoir of biosynthetic genes. Already I discussed this because lots of genes are silent that have the potential to produce bioactive molecules. However, the majority of these genes appear to be inactive for unknown reason. Reason is not known. If we know the reason of this biology, then it would be very helpful to utilize for activating them. Methods to activate these silent clusters would have profound impact on drug discovery. In the earlier section, I already discovered this point. So cryptic molecules can be available. Fermentation maps to improve toxoid production in Clostridia. You can do fermentation as well as the genomic system biology approaches during the fermentation, which genes are expressed and others. And finally, based on new hypothesis, you can design a new Clostridium by genetic engineering, and you can do the fermentation properly to get the enhanced production of the bioactive products. Even in case of biogas plant, we can also do that job, because we can make more biogas, methane gas, by utilizing the bacteria. Toxoid vaccines are used routinely in the livestock. Toxoid means one kind of vaccine. So that vaccine production also can be done through the fermentation of materials by utilizing the Clostridium bacteria. So toxoid vaccines are used routinely in the livestock industry to prevent animal disease caused by pathogenic Clostridia. Vaccines are produced using batch fermentation process, which have undergone limited optimization over the first 5 decades. Low tires and frequent batch failures greatly affect capital utilization and represent a significant cost factor. And to make it profitable, in Queensland University and many other places, projects are going to use high throughput chemistry, omics technology, to produce molecular maps of fermentation. That means every steps of fermentation, if you can, map, then if you know the involved gene very precisely, then you can design a new generation of superior fermentation process. That means fermentation process can be more efficient, and you can produce more toxoid, uh, antibiotics, vaccine, not only toxoid, in many other vaccines which are involved in fermentation process, you can do that. So this is a fundamental, insight to you how high throughput chemistry, system biology can improve the fermentation technology for achieving the higher efficiency in fermentation to produce, um, uh, economically valuable product. So precision nanomaterials based on biomolecule templating. This is another interesting area. So precision agriculture, perhaps you heard about it. So by using nanomaterials, nano emulsion, you can precisely, monitor the peptide and other molecules produced, and silica nanocapsules, you see, put the nanomaterials on bio-inspired silification. So nanomaterials, for example, a certain peptide is a nanomaterial, and then you can make it in emulsion, and then finally can be encapsulated by the silica particle. And then its storability can be increased, and you can make biosensor by using this type of technology, and you can use it for making slow releasing fertilizer and other things, or important drug for targeting the organism specific pathogen or a specific target. Engineered nanomaterials have attracted significant research interest during the past decades for various applications. While compared to man-made materials, precision nanomaterials inspired by nature promise to revolutionize many sectors, including photonics, coatings. For example, some agriculture product, you can make nano coating to increase the shelf life, or nanomaterials, if you can make nano coating like fertilizer.
Like nanofilm.
Can you hear me?
Yes, sir. Sir, coating means, sir, nanofilm.
Nano?
Nanofilm.
Yes, it is like nanofilm on the surface. So
Yes,
healthcare, because of their sophisticated structure, biocompatible core shell nanocapsule have been developed in many labs using a patented technology, the emulsion and biomimetic dual templating platform technology. This technology is based on novel design of biofunctional peptide and protein, acting as not only biosurfactant. Yes, You can make biosurfactant as pesticide, or in the detergent uh, uh, for better washing of your clothes, and also biomineralizing agent by modularizing a partial sequence encoding surface activity with a series of amino acid having biosilification, because some amino acid can make a silification with the the amino acid. Oil core silica nanocapsules are produced under mild condition, including room temperature, neutral pH, without use of any toxic chemicals. We also many scientists demonstrated the facile loading of active or directly dissolving them in the oil phase. So you can dissolve them in oil and make emulsion, and this process help biosilification. That means silicate can be added to your active ingredient. Slow release of hydrophobic ingredients encapsulated in the oil phase can be controlled by tuning the shell thickness. This is telling the how you can make silica nanocapsules. This technology opens a new facile and environment-friendly strategy for fabricating capsules for application in biomedical or agricultural domain, such as slow release of active compounds, for example, fertilizer or pesticide, drug delivery with sustained release. And lots of projects are available for making various nanomaterials built upon this novel technology for slow release and triggering release of active components in biomedical application in agriculture. So this is one of the important point I would like to mention here, that most of the nanotechnologies now discovered and seem useful in agriculture, they are patented. Due to patenting, and not yet large scale production of low cost product, still the application of nanomaterials in agriculture is very limited, but it is a very important area for exploration. That means research, as well as high promise to apply in future for promoting the precision agriculture. I'm telling you maybe the last topic, the engineering striga production in microbes. Striga, perhaps you heard about this hormone, plant hormone, striga. It is associated with the cell elongation, and this is why striga is associated with dwarfism of the plant. And striga is biosynthesized by plant, and scientists now thinking to produce this striga by using microbes. So striga class of plant hormones belong to the isoprenoid family of natural product. Isoprenoid, terpenoid family. Terpenoid is the largest class of secondary metabolites in the natural product area chemistry. They have, even in the plant, it is the largest group of secondary metabolites. They have broad potential as an agricultural chemicals to modify plant architecture. If you want to make a plant, dwarf, that means short in structure, change the architecture, then you can use the striga at certain dose. And a promoter suicidal germination of parasitic seedweed, because striga is involved in germination of striga. Striga, is one of the parasitic weed. So if you use the striga then you can, stop, enhance the germination of the parasitic weed when you have no crop in the field, and then you can kill the parasite. The family is large and chemically diverse. That means striga not a single chemical. There are huge chemical structural diversity, but our understanding is still poor. reconstructing biosynthetic pathway in microbes from the plant to microbes will allow us to examine biochemical pathways and chemical diversity. How many kinds of strigalectones can be produced by changing a little bit of the biosynthetic pathway, then we can get lots of diverse strigalectone and can study the activity of them. So create a new chemical diversity, produce sufficient amounts of study of the physiological effects of different family members of the strigalectone, and ultimately produce sufficient amount of agricultural application. So strigalectone, you see, this is, can you see, this is the striga, and this is the a crop plant. So striga plant always, through Hostoria, they infect the root of the crop plant and get the nutrition. And this parasitic plant, when infect, then plant cannot grow. This is really a big problem in some cases. But if you design a microorganism which is containing the strigalectone producing microorganism, then all the seeds of striga acetica will be germinated before the crop, and they can allow plants to grow without striga acetica. And strigalectone also enhance the production of a growth performance of the plant. So if you want to make dwarf plant, then you can use the strigalectone. So it has high promise in agriculture. So strigalectone is now one of the important subject of plant physiology. Lots of scientists are studying because this compound produced by the plant and enhance the germination of the parasitic weed. So this is a very problematic correlation. It is called chiromone. That means the plant produced by the plant, compound produced by the plant, helping the parasite, which finally infect the plant and damage the plant. So this compound, in true sense, in natural system, are harmful for plants, although plants are producing. Now scientists are trying to understand the biology of the diversity of this compound, chemical diversity, and using system biology or genetic engineering to transfer into microorganism, because this compound is very slow, low concentration in the rhizosphere. So if you use a little higher concentration, all the striga acetica seed can be germinated, and you can kill them before the crop cultivation. So you can clean your land by using this technology, but you need a higher amount. So genetic engineered microbes can be utilized in that case. So this is all about today's lecture. Now let us discuss a little bit with you, and if you have any question, please ask me.
Sir, liptic molecules.
Rana?
Sir, cryptic molecules টা বেশি বুঝি নাই স্যার।
Cryptic?
Cryptic molecules.
Cryptic molecules mean,
Molecules.
there are genes for biosynthesizing or producing that molecule in the genome of a microorganism or plant, but those genes are silent, not expressed. As a result, gene product is the molecule. For example, an antibiotic. So those molecules are not produced. This is why that they are called cryptic molecule. That means organism has the gene. Somehow, if you can trigger the gene to express, then you can get the molecule. This is why they are called cryptic molecules. Somehow they are hidden. They are not produced. So we cannot get those molecules readily, and until and unless we trigger somehow the expression of those silent genes. So lots of the genes are present in an organism, but many genes are silent, not expressed. So even in our case also. So if you can, you know, uh, intentionally trigger those beneficial gene that will give you the cryptic molecules, which may be potentially useful for us as drug or agrochemical, that would be the target of nano biotechnology using the system biology approach. That means to enhance the expression of the gene by using some nanoparticles. Good question. Any other question, Rulamin Rana?
স্যার আরেকটা হচ্ছে স্যার বায়োসিলিফিকেশন
Biosilification. Yes, biosilification means, you know, some amino acids or peptides or an antibiotic or a pesticide active ingredient, when you use directly to the environment, there is a high possibility of loss of that molecule, because pesticide, herbicide that we are using nowadays, a large proportion, more than 95%, are absorbed by the environment, and they are volatile, they are leached out, run off, various losses, for example, urea fertilizer also. So biosilification means by using a biological technique, if you can add silica molecule around the active molecule like amino acid or peptide or urea molecule, and make encapsulated, that means a coating around the particle of urea, and then that silica not, they are abundant in the nature. Silica molecule can allow the active ingredient to release slowly to the target. As a result, efficiency of the bioactive molecules or active ingredients we can significantly increase. And this is one of the process of making encapsulated nanomaterials. Good question. Thank you. Any more? Have you received this file,
স্যার
Rana?
yes sir
Yes, I sent you through email because as you are a single student, I am not using a Facebook group. But for other courses, I always use a Facebook group to share all the files. Today, I shall share one of the interesting recent review article, which is very, nicely or comprehensively reviewed the nanomaterials which are promising in agriculture or agricultural sciences, and published in a Springer book. So please go through that article and step by step to understand the real status, and it is an updated knowledge. I hope you will get huge information. As we wrote, I am also involved in writing that book chapter. So if you find any problem in reading that chapter, please ask me. And you can find lots of our publication in the reference list, and those publications are also available in the internet. If you browse Google Scholar, you can find the link of the PDF link of those articles as original articles, so you can read those. And if you have any problem in access to any literature which you think very interesting for you to study, please ask me. As you are a senior student, master's level student, you have to study the recently published literature. I am giving you some, and you also browse Google Scholar with suitable keyword that you, for example, the nano materials for enhancing the shelf life of horticultural crop or fruit or vegetable, nanomaterials for horticulture. So your homework is, please browse the Google Scholar uh, by using some keywords like nanomaterials on horticulture or nanomaterials in enhancing the shelf life of fruits, nanomaterials for preserving or the vegetables, or nanomaterials in horticultural production. These are the keywords I am giving you homework so that next week you can show me how many literatures you have found. So please list the all published paper and then I can discuss with you whether you can prepare a review article on nanotechnology in horticulture. Because if you can do such an assignment, because you have to do an assignment, if you do such an assignment on nanotech technology in horticulture, different aspects of horticulture, for example, enhancing the shelf life of the fruits, vegetables because they are perishable, as well as the increasing the productivity or in the processing. You can publish that review in a very good journal or even as a book chapter. So I want to engage you in some productive work. So it is your turn uh,
Okay, sir, I will do.
to yes, that would be fantastic. This course, I shall also participate in improving your
Yes,
uh,
sir.
this is why I am giving you today a book chapter. You can understand a structure of the chapter. What are the topics? Even some of the topics in this chapter may be related to your horticulture. Based on that review, you can design your review. But first job, next week, please show me the literature available on nanotechnology in horticulture. So just browse in Google Scholar, nanotechnology in horticulture or PubMed, and list all the papers coming out through the browsing and go through the link and see the abstract first. No need to go through the whole paper and make a reference list and tell me these are the papers published in horticulture which is related to nanotechnology. And it would be very nice because you are a bright horticulturist, and after masters this degree will always be with you. So if you know the involvement of nanotechnology in horticulture, and if you have up-to-date knowledge, and if you can contribute by writing a review, combine all the scatteredly published literature, that would be a good job. Are you agree?
Yes, sir. I'm agree.
Excellent. I will be with you, and it would be a good publication, I am sure. Some of the nanomaterials like Kitusan nanomaterial are found to enhance the shelf life of fruits and vegetables, as well as enhance the pest resistance capacity, that is immune responses of the horticultural plants or other plants. So that would be one of the important molecule, and it also involves in growth promotion. So I am giving you one example of nanomaterial that is chitosan nanomaterial, which is produced from the chitin, the second most abundant natural product. Chitin you can find in the outer shell of shrimp or fungal cell wall or outer shell of crab chitin. And chitin, chitosan is diacetylated product of the chitin. And the chitosan has huge, potential in agriculture as well as in the industry. So please find out some more compounds, nano particles that have already been tested in the horticultural crops. So I am looking for your good effort. Make a big list as much as you can, and tell me brief about the title you collected. This paper describe this paper describe this paper describe this. No need mechanistic, but tell me the overall idea in growth of the horticultural crop. Group the peppers, how many peppers, or enhancing the shelf life of fruits. You can find some of the peppers, enhancing the disease resistance, or the abiotic stress tolerance resistance of the plant. So you can make a small group, enhancing the growth of the plant, or enhancing the shelf life of the vegetables. These are very, very important. And some of them may be used in the horticultural crop as a nano fertilizer. So mm, uh, please work, hard, and it is not so big job, but you can browse Google Scholar and PubMed. These two database is enough for you to get the most of the important publications on nanotechnology in horticulture. Any question regarding this assignment also?
No question. No, sir.
You understood the assignment?
Yes, sir.
Okay, excellent.
If any problem, I will talk you.
Then thank you very much. It is all over today, and this week, you have no more class because what you do, you concentrate yourself for this assignment, and I want to produce something from you. Learning, just memorizing and participating the examination and get some score is not enough for a graduate student. So you are, I hope, an ambitious student, because I enjoyed your field experiment. You are hardworking man. So what you do tomorrow and day after tomorrow and the weekend, you involve yourself in searching the literature. And during the process of searching the literature, if you have any question, please send me an email or call me through phone. Thank you very much.